Thanks. Good. So I'm speaking from UC Davis, and the work we'll be presenting is sponsored by Shriners Hospitals for Children, the Dana Foundation, and National Institutes of Health. So there are techniques now available for universal detection of ASPA mutation carriers and affected fetuses. Unfortunately, they're not universally deployed, and therefore kids are falling through the cracks. You'll be hearing more later on in this session about exciting progress in the development and dissemination of safe and affordable ASPA replacement gene therapy. I'm going to talk a little bit about an interim kind of therapy that may help to bridge the gap until that ASPA gene therapy becomes safe, affordable, and effective. Next slide. So we know that ASPA gene therapy works in mice. Several laboratories, including Dr. Gessler's lab, have shown very convincing data to indicate that that works. But in the meantime, we need more than 1,000-fold scale up to get to humans. Can an alternate therapy be devised that will bridge this gap? A second problem that we have to deal with is that clinical and radiologic abnormalities are already present in most infants and children with Canavan disease by the time the correct diagnosis is made. And in fact, there's some evidence to indicate that by the time a baby is born with Canavan disease, in some cases, the disease is already moved fairly far forward. Can pre-existing Canavan leukodystrophy be reversed? And a sort of conceptual problem. How does a disorder that is primarily in oligodendrocytes, which are the myelin-forming cells in the brain, which are lacking aspartoacylase, which is the protein encoded by ASPA, how does that cause brain vacuolation, particularly inside astrocytes? It's a little hard to conceptualize. Next slide, please. So on your left is a MR image of a child with Canavan disease. And the pale temporal lobes on the two sides are edematous. And actually, more recent magnetic resonance imaging techniques indicate that there's fluid accumulation in the affected areas in Canavan disease. So that those white areas inside the brain are areas that are waterlogged. Cytotoxic edema is the term. Now on your right, you'll see a magnetic resonance spectroscopy spectrum, which can be done in the intact child and measures the level of N-acetyl aspartate, NAA. I'll call it NAA in future. NAA is an amino acid that is broken down by aspartoacylase. Aspartoacylase breaks it down into acetate and aspartate, and oligodendrocytes use the acetate and aspartate to make myelin and keep themselves healthy. So in the absence of aspartoacylase in oligodendrocytes, the NAA levels ramp up in the brain. They get quite high. They may even go to more than double normal levels. Next slide, please. So this just shows an electron micrograph of the brain of a child with Canavan disease. On your left, the top shows sort of holes in the brain. The bottom on the left shows these swollen cystic areas inside astrocytes where the arrows are. So the astrocyte swelling is a characteristic feature of Canavan disease. And on your right, there's a the myelin, which is this multi-layer, it looks like a book, a multi-layered book. There are holes developing within that myelin. So that's intramyelinic edema. So those are characteristics of Canavan disease. How does deficiency of aspartate cause that? Next slide, please. Well, I thought I'd take a minute to just talk about how N-acetyl aspartate makes its way around the brain. And this is a conceptual diagram. There's some parts of it are very well established. Other parts of it are still need more shoring up. But N-acetyl aspartate is made in neurons. There's an enzyme in neurons called NTSAD that pumps out N-acetyl aspartate. The N-acetyl aspartate is released from the neurons and at least in part is taken up by astrocytes, which is that funny looking green cell in the middle. So it flows from the neuron to the astrocyte and somehow from the astrocyte to the oligodendrocyte. And we've shown conceptually that it's going through what are called gap junctions, which are actually holes that in the membrane of the two cells that are joined together by a kind of a tube. But that's something that still has to be fully proven, that those tubes exist, but whether NAA moves through them still needs to be proved. But anyway, this is the way, this is a kind of a plumbing diagram for how N-acetyl aspartate makes its way from the neuron to the oligodendrocyte. Well, what happens if and from the astrocyte to the oligodendrocyte, what happens if the oligodendrocyte can't metabolize NAA because it doesn't have any aspartate, any ASPA in it? Well, like in a plumbing situation, you could imagine that you get a backup of N-acetyl aspartate, and this backup appears to happen particularly within astrocytes and also within myelin sheaths. The next slide, please. So using that conceptual diagram, we asked the question, what happens if you suppress NAA synthesis? And we used a Canavan model mouse. These are mice that are aspartate deficient, and they also develop motor deficits, and their lifespan is decreased. And when you autopsy them, they have pathologic changes like Canavan disease. They have this vacuole or leukodystrophy. So we asked the question, well, what happens if we block N-acetyl synthesis? And we started off with some genetic manipulations, but then subsequently we developed a Antisense oligonucleotide, an ASO. I'll call it an ASO from now on, which when it's administered into the spinal fluid of these mice, shuts down N-acetyl aspartate synthesis, or at least inhibits it to the extent that you no longer get an increase in NAA levels in the brain. And we found that if we gave this to young adult mice that were severely affected by Canavan disease, they recovered. Their motor deficits diminished, they came back to normal. And when we autopsied these mice, they no longer had cystic degeneration in their brains. Next slide, please. So we concluded from that preventing brain N-acetyl aspartate overload by inhibiting NAA synthesis can reverse leukodystrophy in Canavan disease model mice. We were very heartened by this, not only because we like the mice, but because that sounds like maybe in patients who have Canavan disease and have been picked up after they've already developed some features of the disease, motor deficits, sensory deficits, slow development, that some of these features can be reversed. Obviously, the best way to reverse it would be to replace ASPA. And you're going to hear more about ways to replace ASPA. And that certainly works in the mouse. And hopefully in the future, it's going to work in humans as well. So we think that this therapeutic approach may be valuable until and even after effective, safe, and affordable asparagine therapy becomes available, because it's very fast. The antisense oligonucleotides, they may be hard to pick out which ones to use, but after they've been picked out, they're very cheap compared to the cost of developing adeno-associated viral vectors to deliver ASPA to the brain for gene therapy. So in brief, that's my presentation. I'm open for questions.
Well, great. With that, we are super ahead of time, so we have lots of time for questions for Dr. Pleasure. Again, a reminder to our participants that you can type questions into the question and answers box on your screen. If you would like to do that, if you're having difficulty finding it, please send a chat to me or to our producer, Jennifer. In the meantime, I guess, Dr. Pleasure, I can ask you a question to start. So what are your next steps, and what do you plan to do next with this, with studying this technology?
Well, the antisense oligonucleotide that we developed for the Canavan mice works very well for the particular genetic sequence of mouse NTSAD, which is the enzyme that's synthesizing NAA. But it's going to have to be modified in order to work equally well for the human NTSAD. So one of my lab partners, Yan Wang, has a grant in which she is working to optimize the antisense oligonucleotide for human use. I should say a little bit more about antisense oligonucleotides. The ones that we're using have been extensively derivatized so that they have a relatively long duration of action. And these very extensively derivatized antisense oligonucleotides have a nickname. They're called gapmers. And once they're delivered into the cerebrospinal fluid, they penetrate into the brain very easily and into the cells that we want to affect very easily. And in fact, some of these gapmers designed for other genetic diseases are now FDA approved and in use for other rare diseases of infancy and childhood. So we certainly would hope to see that some company or some academic institution or ourselves are able to move forward to develop the therapy for humans. One point I'd like to make there is that working with a mouse is very useful, but to ramp up to the human, it would be very nice to have an intermediate experimental model animal with a bigger brain, not as big as a human brain, but much bigger brain than a mouse, and a brain that's folded more like a human brain is, so that we could be sure that we get the penetration that we want into the whole brain. And it's very heartening to have read just a few months ago that there's a publication from Japan mentioning that they have found Canavan disease cats in their population. Now, unfortunately, cats tend to be spayed. So what's going to have to happen is that they're going to have to go back and find carrier cats and set up a colony. And those cats should be a very valuable intermediate step, not only for antisense oligonucleotide treatment, but also for to fine-tune the asparagine therapy.
Great, well, thank you. And we have had a couple other questions come in from some of our families. One was asking about side effects or potential downsides to this type of therapy, and also asking how often the mice need to receive the ASO therapy.
Yeah, that's the major downside, is that the antisense oligonucleotides, even when they've been extensively derivatized, don't last more than 3 to 6 months. So one would have to give the therapy every 3 to 6 months, which is what's happening, for example, with spinal muscular atrophy, which is another disease in which a antisense oligonucleotide therapy is being delivered. So you would have to give it either by a spinal tap every 3 to 6 months, or by installing a little reservoir so that it could be delivered without having to do a spinal tap each time. That's the main downside of these antisense oligonucleotides. We haven't seen any toxicity in the mice. You want to be sure that the antisense oligonucleotide you're using is not affecting other vital proteins synthesis in the brain or elsewhere. And that's requires doing some rather extensive surveys. But once those surveys are done and you don't find evidences of non-specificity, they tend not to be toxic.
Great. And another question came in asking if this therapy would be helpful for patients who are unable to be treated with gene therapy, either because of their age or disease progression. So could this be potentially used long-term in people who are not candidates for gene therapy?
When the disease is far progressed, when, there's not a lot of literature on this, but when the disease has progressed for years, there's some neuropathologic evidence that neurons start to get lost. I think once neurons start to get lost, the level of reversibility is going to be less. We have some data using these antisense oligonucleotides that early neuronal changes can also be reversed. Not only the cystic change, but the early signs of degeneration in neurons can also be reversed. But I think when you've gone too far, you're probably in a state where this is not going to be valuable, and maybe even asparagine therapy is not going to be valuable. But I think the major roadblock right now to asparagine therapy is going to be, safety and cost, and obviously efficacy. And we're going to hear more about that. I'm very excited to listen to the other speakers to hear about advances along those lines.
Great. Well, I don't think that we have any other questions for Dr. Pleasure right now. So Jennifer, if you'll stop sharing Dr. Pleasure's slides, please.